Picture this, a single molecule holds the key to everything alive today, you, me, the trees outside. DNA and RNA, the blueprints of life, are mind-blowing marvels. But have you ever wondered how they popped up on a chaotic, newborn Earth? Buckle up, this gets wild. Way back in 1871, a guy poking around in pus cells found something odd, a nitrogen-phosphorus mash he dubbed nuclein. Strange start, right? Then, in 1953, everything changed. Two bright minds, with a huge assist from some X-ray genius, cracked DNA's twisty double helix secret. It's like the universe handed us a treasure map to life itself. But here's where it gets stranger. DNA and RNA aren't simple, they're built from nucleotides, little Lego pieces made of sugars, phosphates, and bases like adenine or guanine. These don't just fall together by chance. So, how did they show up on a planet with no labs, no blueprints, no nothing? Let's start with carbon. It's everywhere back then, floating around as carbon dioxide. But turning it into life's building blocks? That's like asking a rock to dance. It's possible, but the moves are tough. No handy catalysts, no perfect conditions, just a stubborn gas refusing to play nice. Methane might have been lurking in Earth's shadier spots, whispering possibilities. Picture a swampy, reducing vibe, cool, right? But coaxing it into complex stuff was a nightmare. Low yields, messy results, it's like chasing smoke with your bare hands. Now, imagine the air thick with nitrogen. Sounds promising, but it's a sleepy giant, super inert. Scientists have tried zapping it into ammonia, the reactive stuff nucleotides crave, but it's a slog. Barely a trickle comes out. Without that spark, we're stuck. Phosphorus is the real tease. It's crucial for nucleotides, but on early Earth, it's hiding in rocks that won't budge or sprinkled in by rare meteorites. Imagine waiting for a cosmic delivery truck that barely shows up. How's that gonna fuel life's kickstart? Shift your gaze to the skies, lightning rips through, UV rays blaze down with no ozone to stop them. These could have sparked tiny miracles, cooking up formaldehyde or hydrogen cyanide, seeds of bigger things. It's a cosmic kitchen, but the recipe's tricky. Down in ancient oceans or steaming vents, those seeds might have sprouted. Evaporation could have concentrated them, minerals might have nudged them along. But the Formose reaction, supposed to churn out ribose sugar, spits out a gooey mess instead. Where's the precision? Energy's the wild twist. Lightning zaps, vents bubble, radioactive rocks hum, asteroid crashes shake things up. Each could ignite chemistry, but here's the catch, they wreck stuff too. UV builds molecules, then blasts them apart. It's a creation-destruction dance, beautiful, but brutal. Now, nucleobases, adenine, guanine, cytosine, uracil, these are the alphabet of life. Making them? A headache. Adenine needs insane amounts of cyanide, way more than Earth could muster. Guanine's a ghost, showing up in pinpricks after Herculean effort. Cytosine and uracil aren't much cheerier. Cytosine's a no-show in lab sparks and falls apart fast, think 340 years tops. Uracil forms under heat, sure, but at 100 degrees, it's got 12 years before it's toast. How do you build a library with pages that burn? What if they hitched a ride from space? Meteorites like Murchison drop nucleobases, cool, right? But surviving cosmic rays and crash landings is a gamble. Plus, the hull's tiny, not enough to rewrite the story. It's a tantalizing clue, not a solution. Here's the jaw-dropper, out of a bazillion possible molecules, think gazillions, life picks just 5 bases. Adenine, guanine, cytosine, uracil, thymine. No guide, no coach, just blind luck. That's like winning the cosmic lottery with no ticket. Water's the ultimate paradox. It's gotta be there, reactions need it, but it's a wrecking ball too. Nucleobases crumble in it, adenine's gone in 80 years at mild temps, guanine's toast in months when it's hot. How do you stack the deck when the table's underwater? Stability's another gut punch. At 100 degrees, adenine lasts a year, guanine 10 months, blink and miss it stuff on Earth's timeline. Chill things down, they last longer, but then nothing forms. It's a cosmic catch-22, build fast or rot slow. Ribose, the sugar star of RNA, is a diva. It's perfect, flexible, fits like a glove, but making it? Sugars like ribose aren't just sweet, they're the backbone of life's blueprints, RNA and DNA. But how did they show up on a wild, young Earth? Buckle up, this ride's a cosmic puzzle. Ribose is the star player here. It's the 5-carbon sugar holding RNA and DNA together, letting them store life's genetic code. Its neat ring shape fits like a glove, but making it back then? That's a mystery scientists can't crack easily. What seems impossible today might rewrite tomorrow's science. Why's ribose so special? It's got a unique structure, stable yet bendy, perfect for nucleotides. Over 280 other sugars got tested as stand-ins, but none worked. Ribose rules because it nails homochirality, life's right-handed twist. How'd it win that cosmic lottery? Now, imagine a messy kitchen on early Earth. The Fermose reaction, cooking sugars from formaldehyde, sounds promising, right? Found way back in 1861, it's the go-to idea for ribose's origin. But here's the twist, it's a chaotic chef, and ribose is just a crumb in the mix. This reaction's a wild party, formaldehyde piles up into sugars under steamy, alkaline vibes. It could have sparked ribose, sure, but the yield's tiny, less than 1%. You get a gooey soup of dozens of sugars, not a clean batch of life's key ingredient. What a tease! Ribose isn't simple, it's a shapeshifter. It flips between right-handed and left-handed forms, plus alpha and beta styles. For setups, alpha D, alpha L, beta L, and a flexi ring that twists. These quirks make it king of nucleic acids, but a prebiotic nightmare. Life picks beta nucleosides, where ribose and bases bond cis-style, keeping things tight. Alpha versions? Rare flops with a trans setup. Only right-handed beta-deribonucleotides rule, locking the helix steady. How'd nature pick that winner without a coach? But here's where it gets stranger, ribose is a diva. It forms in that Fermose mess, but vanishes fast in water. No clear recipe makes it stick around, and its right-handed chirality? A total fluke in a random world. Borate minerals might help, but were they even there? Gerald Joyce wrote a science paper which was published in Nature magazine, in 1984. His findings suggested that in order for life to emerge, something first had to crack the symmetry between left-handed and right-handed molecules, an event biochemists call breaking the mirror. Since then, scientists have largely focused their search for the origin of life's handedness in the prebiotic worlds of physics and chemistry, not biology, but with no success. Phosphorus joins the fray, it's the glue for nucleotide backbones. On early Earth, it's locked in apatite rocks, barely trickling out. Oceans had zilch-free phosphate, precipitation snatched it up. How'd it break loose to kickstart life's chemistry? Let's wrap this up with life's earliest roadblocks. Carbon sources were tough, CO2 reduction was a thermodynamic mess, and methane was no better. Its utilization gave pitifully low yields, barely scratching the surface of what life needed to form complex molecules. No prebiotic carbon fixation existed to help. Nitrogen? A struggle too, dinitrogen fixation with its triple bond took too much energy, and ammonia wouldn't stick around. Phosphorus stayed locked in stubborn minerals, even with meteoric delivery. Oxidation didn't help. Wild. Nucleotides, how did they come together with no labs, no enzymes, just raw chaos? Buckle up, this gets mind-bending. Phosphorylation's the magic trick here, adding phosphate to nucleosides to make nucleotides. Today, enzymes nail it, but back then? No such luck. How did this energy-hungry process kick off on a planet with no rulebook? The answer might rewrite everything. Imagine sugars getting jazzed up with phosphate, then teaming with nucleobases to form nucleotides. Tidal pools drying out, geothermal shores sizzling, these spots might have cooked it up. With stuff like formaldehyde around, it's a tantalizing hint at life's first steps. But here's the twist, phosphate's a loose cannon. It's super reactive, bonding with anything it bumps into. Without enzymes playing referee, you'd get a mess of useless molecules, not the neat nucleotides life needs. How'd it pick the right dance partner? Scientists have tossed out wild ideas to crack this. Trimetaphosphate, think phosphate on steroids, might have sparked reactions. Cyclic phosphate's another contender, popping up in hot vents or volcanic zones. Could these fiery spots have been life's kitchen? What if regular nucleotides were too tricky? Some say alternative versions, with funky sugars or tweaked phosphates, might have stepped in first. Easier to whip up, maybe, but they still needed phosphate magic and stability to kickstart life's party. The inevitable conclusion of this survey of nucleotide synthesis is that there is at present no convincing, prebiotic total synthesis of any of the nucleotides. Many individual steps that might have contributed to the formation of nucleotides on the primitive Earth have been demonstrated, but few of the reactions give high yields of products, and those that do tend to produce complex mixtures of products. In 2017, Sutherland formed nucleotides, amongst all of the basic building blocks, lipids for compartments, amino acids for metabolism, starting with cyanide as a common initial substrate. However, this ended up requiring 6 separate ponds with their own unique geochemical conditions and whose products then needed to be mixed together in a specific sequence. Even this degree of complexity did not supply required products, but only precursors. The requirement of so many unique ponds and associated chemistries in such close proximity to each other, of course, stretches plausibility. And Graham Cairns-Smith noted, I think, to say that on average the 14 hurdles that it would take to make primed nucleotides would each take 10-unit operations, that at least 140 little events would have to be appropriately sequenced. Unguided, the appropriate thing happened at each point on one occasion in 6. The odds against a successful unguided synthesis of a batch of primed nucleotide on the primitive Earth would be a huge number, represented approximately by a 1 followed by 109 zeros, 10 to the power of 109. How did nature start to play this game? At the very least a maintained supply of primed nucleotides would be required for any kind of organism using our kind of message tapes. A nucleotide-making factory would be needed. The odds are enormous against its being coincidence. No figures could express them. Now, imagine linking nucleotides into chains, phosphodiester bonds tying them tight for DNA and RNA. Enzymes make it a breeze today, but prebiotically? Wet-dry cycles or clay minerals might have nudged it along. Still, we're stuck with puny chains, too short for life. Clays like Montmorillonite could have been MVPs, sparking RNA oligomers with their silicate aluminum vibe. But they're picky, source matters, and they need tweaking to shine. Even then, you get 40 nucleotide stubs, cool, but not enough for the big game. No known methods enable the thermodynamically unfavorable reaction of forming nucleosides in aqueous solution. While dry-phase synthesis has produced purine nucleosides, it fails for pyrimidine bases and ribose. Laboratory syntheses of ribonucleotides rely on protecting groups to overcome thermodynamic and kinetic barriers, requiring controlled manipulation of sugars and nucleobases. Energy's the big snag. Phosphorylation guzzles power, and early Earth had no ATP handouts. Tidal pools or vents might have sparked it, but conditions flipped fast. How'd nucleotides form steady in that roller coaster world? Prebiotic processes are similar in character to dumping a tank of gasoline on a car and igniting it. By contrast, living cells have machinery which converts energy appearing in a specified form into ATP, the energy currency of the cell, which is useful for biotic processes. Summarizing, phosphorylation needs energy, yet prebiotic Earth lacked high-energy molecules. Even if phosphate was present, it wasn't reactive enough, making its source uncertain. And if nucleosides did get phosphorylated, they risked forming useless byproducts. What if I told you that the building blocks of your DNA hinge on a chemical trick that's been puzzling scientists for ages? Let's unravel this mystery together. First, let's break down what ribonucleotide reduction even means. Imagine a ribonucleotide. It's like a tiny Lego piece with an oxygen atom attached to its ribose sugar. Now, this process strips that oxygen away, turning it into a deoxyribonucleotide, the stuff that makes up DNA. Sounds simple, right? But here's where it gets tricky. This reaction is a big deal, and scientists are still scratching their heads over how it happened in the chaotic early Earth. Now, let's talk about the star player in modern cells, the ribonucleotide reductase. This enzyme is like a master chef, carefully controlling the redox reactions that make this transformation possible. It's efficient, precise, and keeps everything in balance. But here's the catch: back in the prebiotic world, before life as we know it existed, there were no enzymes. So how did this reduction happen in a world with no biological helpers? That's the million-dollar question. Picture the early Earth, a harsh, wild place with no life, just raw chemistry at play. The lack of enzymes in this prebiotic environment throws a massive wrench into the story of ribonucleotide reduction. Without those biological catalysts, how could this reaction even get started? Scientists are stumped, because the energy needed to pull this off in such a barren setting is a real head-scratcher. But don't worry, we're about to explore some wild ideas that might just crack this case. One theory suggests that non-enzymatic pathways, like chemical reducing agents, could have kicked things off. Think of these agents as nature's stand-in for enzymes, nudging the reaction along without any biological help. It's a clever idea, but here's the twist: where did these agents come from, and how did they work in the messy, unpredictable conditions of the early Earth? The plot thickens, and we're just getting started. Now, imagine a high-energy environment. Think volcanoes erupting, lightning slashing the sky, and hydrothermal vents bubbling with heat. Some scientists believe these extreme conditions might have been the perfect stage for ribonucleotide reduction. The intense energy could have driven the reaction, but here's the challenge: how do you control that chaos to make something as precise as a deoxyribonucleotide? It's like trying to bake a cake in the middle of a storm, possible, but tricky. Ribonucleotide reduction is a cornerstone of life, but its origins are shrouded in mystery. From the enzyme-driven precision of modern cells to the chaotic, high-energy possibilities of the early Earth, this process challenges everything we know about chemistry and biology. Could UV light or chemical agents have sparked the first steps toward DNA? Or is there another piece to this puzzle we haven't found yet? No enzyme blueprints, no metabolic maps, chirality's a fluke, energy's a ghost, and phosphorylation's a stretch. Nucleotides staying put in water? Fat chance. Prebiotic protocells pulling it together? That's a leap science can't land. Ribonucleotide reductase cuts oxygen like a pro today. Prebiotic hacks couldn't match it. UV or vents might have tried, but selectivity tanked, and reducing agents were iffy. Stable deoxyribonucleotides in that soup? Good luck. From sloppy prebiotic brews to cells churning out nucleotides, it's a chasm. Catalysis, info transfer, energy tricks, and cell walls, none click without a push. Life's simplest setup still towers over Earth's raw chaos. This dive into nucleotide origins leaves us reeling, prebiotic hurdles stack high against modern finesse. From phosphorylation flops to DNA's rise, the gaps gape wide. Could chemistry alone bridge it, or is something bigger lurking? Thanks for riding this cosmic wave. Nucleotide formation's a riddle from Earth's dawn to now, hit us up to dig deeper. The universe isn't done teasing us with life's wild start, stick around for more. Thanks for watching. If you enjoyed the video, don't forget to like, subscribe, and hit the notification bell so you never miss an update. Let us know your thoughts in the comments below, and stay tuned for more exciting content. See you in the next one.
Hey naturalist, you think life's a random roll? Let's break it down, this story's got a soul. Picture early Earth, a wild chaotic brew. No labs, no maps, just a chemical stew. Nucleotides for DNA, they're the key to the game. But making them from scratch, that's a losing claim. Carbon stubborn, methane shy, nitrogen won't wake up, no matter how you try. Phosphorus is hiding, locked in rocky stone. How's this cosmic soup gonna build a home? No chance, it's a plan, can't you see the design? Life's too tricky for chaos to align. From robos to bases, it's a perfect fit. Intelligent creation, God's the one who lit the hit. Ribose sugar's a diva, Formose makes a mess. Less than 1% yield, nature's under stress. Chirality's a fluke, right-handed all the way. No coach, no guide, random luck, you say. UV zaps, lightning cracks, energy's a beast, but it builds and breaks, oh what a feast. Nucleotides crumble, water's a wrecking ball. Adenine's gone in a year, how'd life start it all? No chance, it's a plan, can't you see the design? Life's too tricky for chaos to align. From ribos to bases, it's a perfect fit. Intelligent creation, God's the one who lit the hit. Phosphorylation's a puzzle, no enzymes in sight. Tidal pools and vents can't get it right. Out of gazillions, just 5 bases win. Blind luck or a blueprint, where do we begin? The Bible says seek and you will find. Matthew 7:7,